Hi. Hi, everyone. Thanks to the organizers. I'm very grateful to be here and very excited to present our research. I have to quickly correct that I'm now a PhD student at the University of Tübingen and in Jakob Mackes' lab. And we are collaborating with Srini Turaga, and they both jointly supervise me on this project. Okay, so machine learning provides successfully models which perform complex, challenging, meaningful computations. And computation neuroscientists often deal with models that are biologically realistic. And for us, the overarching question is how can we construct models that are both performing meaningful computations and are biologically realistic. And supporting our research question is this finding that computation neuroscientists can use deep convolutional neural networks that are only trained to detect objects, to explain neurotuning in higher visual areas of the monkey and the human. And this was first published by Dan Yemens and James DiCarlo in 2014, and I took these nice illustrations from the review. I also want to mention that I'm really looking forward to this fantastic work and the keynote lecture from James DiCarlo today. However, one thing that these convolutional networks are lacking is a mapping of artificial neurons to biological neurons, and they often feed forward and don't have temporal dynamics. So what we want to do is to constrain basically CNNs and our models by a structure of the circuit, so the actual neural circuit structure. We want to equip the neurons with temporal dynamics, and we want also to use task optimization and task constraints to make these networks then perform challenging computations. And we will do this, we can do this for the fruit fly visual system. And the ethologically relevant task for the fruit fly is to detect motion. All right, let's first look at how we can constrain the CNN with the connectomic data. And briefly, the fruit fly nervous system contains about 250,000 neurons, from which 150,000 neurons are visual system neurons. And the cool thing about the fruit fly is that we have specific neuron types. We basically typed all the cells in the visual system, which allows us to precisely map circuit structure to function, and that is consistent across fruit flies. Okay, and to constrain CNNs with the connectomic data, we use the hexagonal arrangement and columnar architecture of the fruit fly visual system. So here you can see that the photoreceptor cells in the retina of the fruit fly are arranged hexagonally in these visual columns. And this hexagonal arrangement also is preserved downstream in terms of the axons and the dendrites, which build these distinct subregions of the optic lobe, the visual system of the fruit fly. And now on the right-hand side, you see that our model resembles this hexagonal architecture, and that here, each regular hexagon represents one cell type, and each tiny hexagon within is one individual cell. And those lie within the different subregions of the visual system. And we have in total 65 cell types that we model, 44,000 individual cells, 1.5 million connections in our model. Okay, but how do we actually know what the connections are that we need? And for that, Fabian Schopp actually derived these convolutional filters that you see in the bottom left from dendrites with synapse annotations. And these convolutional filters represent the connectivity between a pair of connected cell types in terms of the synapse count at a specific offset locations between like the visual columns. And we also estimated the signs with a study of neurotransmitter and receptor profiling and help from Ayosha Nur and Janne to say if a presynaptic neuron communicates excitatory or inhibitory onto a postsynaptic neuron. And in total, I illustrate this whole connectivity matrix that we get from it for all the 65 cell types that are almost the entire early visual system of the fruit fly. And this is the connectivity that we use to constrain our CNN. Okay, so now that we have that, we ask, okay, how can we constrain the neuron's state, and model the temporal state and the dynamics and the communication between the cells. And for that, we use a simple mechanistic model of single-cell dynamics. And here in this equation, this describes a coupled system of non-spiking point neuron voltages, because the neurons in the fruit fly visual system are non-spiking and they are small. And in this equation, we don't know the time constants of a neuron type Ti, of a postsynaptic neuron I, and we don't know the resting potentials. In the center, you see the currents, which we describe as simple current-based synapses. They are proportional to the rectified presynaptic voltage. These synapse counts that we know from the reconstruction, the estimated signs that we have, and then we multiply a scaling coefficient to translate to precise units of currents that we don't know otherwise. So in total, our model has 734 parameters, and without a connectome, such a model would have 35 million parameters. So our connectome reduces the number of free parameters that we now have to estimate to 734. Okay, and we estimate these parameters with the task of motion detection, because we know that's ethologically relevant to the fruit fly. So we train the networks to detect motions. And what does this mean? On the left-hand side, we see we use short video sequences as input that we render to this hexagonal geometry of the fruit fly eye. And then this represents the network. So each neuron now implements these set of differential equations. And with a movie input, we can integrate the dynamics of this coupled system using Euler integration, and then we can decode the responses in the network to the optic flow or the motion ground truth that we have. So we have a synthetic ground truth, and then we can compare the prediction to that ground truth and use the prediction error backpropagated through time to update the parameters in the networks, this 734 parameters that we want to find. Okay, here's one example. So left is the video input. Please play the video, if you can. Okay, I guess the animation got lost somehow in a conversion process. Yes. Okay, I'm sorry, the video is not working, unfortunately. So what you would have seen here is that we get a prediction of the optic flow in the trained network. So this is one example of the trained network, which captures, so on the top right, you can see basically one frame, and you see already in this frame that it captures essential features of the motion. It's looking different, though, from the synthetic ground truth. And we also don't know if the fruit fly perceives motion as the synthetic ground truth would imply. But so this is basically the predictions that we can make from one model. But more importantly, we can estimate these 734 parameters and get predictions for neural tuning. And if we now trained a number of models from different random initializations, we find this task error distribution. So on the left-hand side, you see the best model. On the right-hand side, you see the worst model. And this tells us that models basically fall in different local minima. So they find different parameter solutions and tuning solutions. But note that at initialization, the task performance is much worse, which tells us that still models become very good at learning motion detection. And now, to say something about how realistic the tuning predictions are that these models produce, we can do synthetic neurophysiology and compare those single cell types in our model to the ones that are measurable experimentally. And here you can see one example where one measures these characteristic direction selectivity tuning curves of motion detector neurons. And we take from the 50 models that we trained earlier, so that we have seen in the task area distribution, we take 20% of the best models, for which we know, okay, they learn to detect motion reasonably well, and then do such synthetic neurophysiology. This is a video where we see here in the photoreceptor cells the input, which are bright moving edges, moving to 12 different directions. And then here we see basically all the responses of the cells that we model. And we see certain cells, unfortunately we can't see it in this single frame, but we see that certain frames invert the input, so they have certain contrast preferences, and other cells extract the motion, like an edge of the of this visual input. And so we can quantify this for each cell type in the model using the responses to these simple stimuli that we use. And here you see this quantified as on-off selectivity on the y-axis, and values above 0 are cells which prefer if bright flash, and values below 0 are cells which prefer dark flash. And we know for these yellow cells on the left that they all prefer bright flash, and we know from experimental measurements that those cell types in violet prefer the dark flash. And our model, or across the 20% best models, the median of this quantity predicts all these known properties. And maybe more importantly, because this contributes to the motion detection capability of the cells, we quantify the direction selectivity in the models, and values above 0 mean that the cell is direction selective. So it responds strongly to motion in a particular direction. And we know for T4 cells here in red that they are selective to motion of bright things, and T5 cells are selective to motion of dark things. And actually the medians across our best task performing models again predict these tunings. In addition, we hypothesized that these TMY3 and TMY17 cells are also motion detectors, and a collaborator, Nathan Klapatka at Janne, is now preparing experiments to actually test that. I'm quickly summarizing. So we asked, how can we construct models that perform meaningful computation and are biologically realistic? And for that, we built a recurrent neural network model consistent with a fruit fly connectome, and we trained the network to detect motion. And cells in the network acquire the tuning which is consistent with measurements. And our approach implies a way to investigate data-consistent models of neural computation and generates hypotheses for function of uncategorized cell types. All right, thanks to Srini and Jakob, who supervised me, at Mason McGill, and to Mason and Fabian with help on data structures and algorithms, synapse clustering done by Fabian, and feedback of the Marke Lab and Turaga Lab, which was great. All right, this was it. Thanks, everyone, and I'm ready to take your questions.